Here, in the middle of Berlin, at the Charité crossover building, pioneering work on the brain is being done to understand how we learn and remember the details of our lives. From the nuanced neural pathways of fruit flies to the intricate cerebral networks of humans, scientists at the Charité are crossing the frontiers of neuroscience. Important work is going on using different species. That's because scientists have found that each species studied offers unique insights and helps to piece together the vast puzzle of cognitive processes, memory formation, and neural plasticity. This building is a place where multidisciplinary teams converge on groundbreaking discoveries.
Welcome to our lab at the Institute of Neurophysiology of the Charité in Berlin-Mitte. Here we're trying to understand one of nature's most exquisite puzzles: how memories are formed, stored, and consolidated in the brain. Using the fruit fly as our model, we explore the brain's building blocks. These tiny creatures offer us a window into understanding the fundamental processes of memory. Fruit flies have a relatively simple brain structure in comparison to humans, which allows us to observe and manipulate specific memory processes. Since a new generation of flies takes just 12 days, they're perfect for genetic studies.
Our team has made fascinating discoveries about how dopamine signals reward in the fly brain and how this affects memory strength. With our focus on the mushroom bodies, which are important regions of the fly brain for memory, we're uncovering not only how memories are stored, but also how they're dynamically shaped by experiences such as reward and novelty.
We examine flies while they are learning and after they have slept to see what happens in their brains. And it's during sleep that we've observed crucial changes in brain activity. We see that after learning, there's a decrease in synaptic strength or long-term depression at some neural sites, and these are marked in blue. This is contrasted by an increase in synaptic strength or long-term potentiation at others, shown in green. We also see changes in spontaneous activity in some neurons after training flies. And the brain is remarkably active during sleep. Likely, these activity patterns will contribute to memory consolidation, similar to what likely happens in humans.
This weakening and strengthening of synapses in different areas of mushroom bodies are what biases the animal towards a specific direction. It's a neural dance of approach and avoidance, orchestrated by experience.
We've discovered that sleep-related oscillations in specific ring neurons are a marker of sleep drive. The stronger the sleep drive, the higher the slow-wave oscillations. These oscillations are crucial. Suppressing them means lighter sleep and more frequent awakening. Moreover, after such disrupted sleep, we see compensatory increase in sleep, what we call rebound sleep. Our recent work has revealed that in the connectome of the fly, 3 main networks stand out. Using simulations, we've shown that these can sync. Notably, the R5 neurons, which are mainly inhibitory, interact with the excitatory helicon neurons. The rhythmic activity of these neurons, their burst mode, acts as a gate. It cuts off sensory information flow to downstream networks. The windows of upstate remain, and these could be key to waking the fly. Interestingly, these networks allow for slow synchronization over the course of the day, with inputs from the circadian clock and homeostatic signals influencing the synchronization. It's a fine-tuned system, delicately balanced. During sleep, the synchronization pattern can change, and this change is dependent on a fly's previous experience. This provides a direct link between a fly's memory and environmental cues.
Our work aims at understanding neural activity in the context of behavior. We collaborate with a group of Jörg Winter from the Humboldt University to develop, for instance, behavioral assays. Likewise, our work profits from interacting with computational neuroscientists.
Understanding the brain functions that underlie memory consolidation and sleep requires the ability to monitor the right behavior at high precision. In my lab, we developed the experimental stages on which performing flies make visible to us their memory performance or even their tiredness. When these experimental paradigms are then combined with optogenetic research strategies, fly behavior can be remote controlled and simultaneously the brain activity can be read out. This has already allowed us to understand hierarchies between neural networks promoting both locomotion and sleep.
If you look at the neurons of the brain like an orchestra, each neuron an instrument. In the fascinating world of the fly brain, we can imagine some fly neurons are like musicians playing two instruments at once. This is unusual because, typically, a neuron in the brain uses just one kind of chemical messenger or neurotransmitter to communicate. But here we see specialized neurons. There are 5 neurons using 2 different transmitters. In our sleep, the brain shifts into a different mode, like a sports crowd in a stadium starting to cheer in unison. This transition likely allows the brain to consolidate our memories, but we don't understand how. My work involves creating computational models to understand how these fly neurons, with their dual transmitters, help achieve the right conditions during sleep for memory consolidation. It's there setting the stage for the brain to transition into these crucial synchronous oscillations, a mysterious yet vital part of how we learn and remember.
Similar to humans, flies experience transitions in sleep, suggesting perhaps that despite our differences, fundamentals of how brains work might be surprisingly similar. By simulating how this network behaves, we're uncovering the delicate balance that prevents overexcitation and maintains just the right rhythm for a good night's sleep, setting up our memories for the next day. It's a puzzle we're only just beginning to piece together, and it's as exciting as it is important. Our work into the cellular circuits of memory and sleep reveals not just how memories are formed, but how they communicate across brain regions, offering insights into the universal principles of memory consolidation.
From the microscopic intricacies of the fly brain to the vast complexities of human memory, our team is unlocking the secrets of how we remember, learn, and evolve.